Much of our foundational knowledge of immunology stems from mammalian models, specifically the lab mouse. For specialists, trying to map these rigid mammalian frameworks onto avian biology creates significant gaps. Birds and mammals followed parallel evolutionary trajectories. This resulted in two distinct immune architectures that solve the same biological problems through different means. Examining the avian system reveals missing genes and fragmented receptors. However, birds neutralize pathogens effectively, using unique chemical and cellular pathways that fulfill the same requirements. Recognizing these differences is necessary for accurate work. Treating avian immunology as a slight variation of our own can lead to mistakes in veterinary pathology and vaccine design. Every vertebrate requires an innate immune system that acts immediately. It must suppress an infection during the days or weeks it takes for the adaptive system to mobilize. This speed is enabled by pattern recognition receptors, or PRRs. Unlike complex T-cell receptors, these are encoded directly in the germline DNA. Being germline encoded, they are abundant. Millions of innate cells can identify and react to pathogens instantly, without waiting for cellular division. Since birds and mammals share this requirement for instantaneous recognition, we must look at how the avian lineage achieves it, while missing specific genes common to mammals. The innate response relies on pre-configured structures rather than memory. This forced the avian lineage to engineer distinct molecular solutions to survive. When systemic inflammation occurs, the liver secretes acute phase proteins to coordinate the defense. While birds share standard proteins like C-reactive protein with mammals, they lack the gene for a molecule called haptoglobulin. In mammals, haptoglobin binds to free hemoglobin released during red cell destruction, neutralizing it before it causes oxidative stress or feeds bacteria. The avian solution is a unique protein called PIT54. Though from a different family, it performs the exact same defensive role. The liver's use of PIT54 shows that evolutionary pressure will co-opt unrelated proteins to ensure a vital chemical defense remains functional. This adaptability also appears in the complement system, specifically within the structure of collagenous lectins like collectins and ficolins. These molecules are composed of trimeric subunits resembling a bouquet of tulips. Carbohydrate recognition domains at the top allow them to bind to microbial surfaces with high avidity. When mannan-binding lectin attaches to a pathogen, it activates mast proteases, initiating the lectin pathway. This chart maps the three distinct pathways, classical, lectin, and alternative, that trigger the broader complement cascade. Regardless of the initial trigger, the cascade converges at the cleavage of the C3 convertase. This convergence ensures the system triggers essential downstream functions, like opsonization and the assembly of the membrane attack complex, regardless of how the pathogen was detected. On the cellular frontline, birds deploy heterophils in place of mammalian neutrophils. Avian heterophils have a specific biochemical deficit. They completely lack the enzyme myeloperoxidase. Without this enzyme, heterophils cannot execute a standard oxidative burst to produce reactive oxygen species for killing bacteria. To compensate, avian heterophils rely on a heavy deployment of antimicrobial peptides. Birds utilize a suite of 14 beta defensins. These peptides kill pathogens by physically perforating the membrane, forming disruptive pores that cause the cell to fail. This shift in strategy moves the avian primary killing mechanism away from chemical oxidation toward physical membrane disruption. These customizations extend to the cell-associated sensors, particularly the toll-like receptor repertoire. Comparing the avian and mammalian genomes reveals several genetic departures in the toll-like receptor locus. Birds have no ortholog for TLR9, and their TLR8 gene is fragmented by a genetic insertion. Mammals use TLR9 to detect unmethylated DNA motifs, common in bacteria and viruses. Birds manage this same detection using a uniquely adapted sensor called CTLR21. Birds also possess CTLR15, a receptor found only in avian and reptilian lineages. CTLR15 is activated by heat-label yeast agonists and functions differently than any known mammalian sensor family. This bespoke repertoire provides an optimized array of sensors for the avian environment, filling the functional gaps left by absent mammalian genes. Zooming out to the holistic network reveals how unique avian elements like PIT54 and CTLR21 interact with conserved nodes like the C3 protein. The core strategy remains consistent across vertebrates. Use germline-encoded sensors to protect the host before the adaptive system activates. The avian lineage engineered unique molecular pathways to reach these identical endpoints. It traded oxidative bursts for physical pore-forming defensins and replaced haptoglobulin with PIT54. Identifying these specific divergences is the only way to accurately model disease and develop effective vaccines for birds. Vertebrate immune evolution follows a modular path. The mammalian model is one successful architecture among many for surviving in a pathogenic world.